Yeah, welcome to the next part in catabolism. If there is an homofermentative catabolic pathway for lactic acid fermentation, there must be also an heterofermentative one. And this is coupled also to the pentose phosphate pathway. Here we go. Here is how it works. Yeah, you have learned about glycolysis already. You might have already watched about the homofermentative lactic acid fermentation pathway. And the big question is, if there is no aldolase, no glycolysis, what now? Welcome to the world of heterofermentative lactic acid fermentation. And in this case, heterofermentative lactic acid fermentation has much to do with another pathway called the pentose phosphate pathway. So we will talk in this section about the pentose phosphate pathway, and we will talk about in this section about heterofermentative lactic acid fermentation. Here we go. Okay, again, this pathway depends on a full glycolysis pathway. I produce pyruvate, so in this pyruvate then I use as a internal electron acceptor to produce lactate. Now, what happens if I have no aldolase? So there are bacteria around who have no aldolase. So if you have no aldolase, obviously you don't produce glyceraldehyde 3-phosphate. You cannot go down there. And in these cases, bacteria can use another pathway. They can use another pathway, and there we have to do a little excursion to the so-called pentose phosphate pathway. And the pentose phosphate pathway starts differently. So you remember my first step in glycolysis. I start with glucose. Then I have kinase reactions and isomerase reactions producing fructose 1,6-bisphosphate. There is no oxidation happening there. I just have phosphorylated the molecule and rearranged it a bit, and then we split it and go ahead. So on the C6 level, there is no oxidation happening. This is different in the pentose phosphate pathway. So there I start with an oxidation. So here, of course, again, I have my glucose. Now I have the open projection in order to show you what's happening. Of course, in solution, we have this pyranose-cyclic form, but we don't care now. So this is glucose 6-phosphate. And what I do now is I start with an oxidation. And of course, you could oxidize all the hydroxyl groups here. But let's say if you are not sure what is being oxidized, and if you have an at the same time hydroxyl and aldehyde or aldehyde groups, bet on the oxidation happens at the level of the aldehyde group. This is, of course, not something if you have a new molecule in the exam and I ask you, you cannot be sure whether this is the hydroxyl group or aldehyde group. There you will get a hint what to oxidize. But now, let's say in general, it's the aldehyde group. So we could oxidize the aldehyde group. Of course, we could already name the enzyme. The enzyme catalyzing this is an enzyme oxidizing glucose 6-phosphate. So this enzyme must be called, after the citric acid cycle, all of you know it, this enzyme must be called glucose 6-phosphate dehydrogenase. Full stop. That's it. So the first step here is the oxidation of glucose using glucose 6-phosphate dehydrogenase, and we get 6-phosphogluconate. Of course, it's a redox reaction. Of course, we need to reduce at the same time something. We come back to it, and we have oxidized here our aldehyde group into a carboxyl group. And in this case, we have produced NADPH. So this doesn't produce NADH, this reaction, it produces NADPH. So this is also a way to generate reduction equivalents for biosynthesis. Now, we are at 6-phosphogluconate, and the next step, an oxidative decarboxylation. If I decarboxylate, so of course this group will leave. If I have no oxidation, this will of course stay then a hydroxyl group. So what I could do is I could oxidize this hydroxyl group into a aldehyde group. But in this case, what is done is not this group is oxidized. This group is being oxidized, so I come up with a keto group at this C3 position, and this carbon dioxide leaves, and in this case I get ribulose 5-phosphate. This molecule is just called ribulose 5-phosphate. This is the problem with the sugars. They all have different names. And of course, this is something which is a pain in the ass to learn them by heart. I don't expect you to know by heart the structure formula of ribulose 5-phosphate. I expect you to know that the first reactions in the pentose phosphate pathways are 2 subsequent oxidative reaction, and 1 is a decarboxylation. And you see I have here a legend of the number of carbon backbones, and you see you go from a C6 to a C5 carbon here. Okay, carbon dioxide leaves, of course. And then I have ribulose 5-phosphate. So this is the start in the pentose phosphate pathway. So I generate from an hexose now a pentose. Yeah, of course, naming this enzyme is simple. It's a dehydrogenase because it's an oxidative decarboxylation, and the substrate is 6-phosphogluconate. So it's 6-phosphogluconate dehydrogenase. And in the second part, we have couple of reversible non-oxidative reactions. So the pentose phosphate pathway, and this is kind of your initial question, caloric value of things and so on. So in the pentose phosphate pathways, you use isomerases and epimerases. You remember isomerases can transfer the keto group, for example, to aldehyde group, or move it down. So from the ribulose 5-phosphate, where you have the keto group at C2, you just shift it to C1 and you get ribose. And of course, you remember ribose is important for RNA, and if you deoxidize, it's DNA and so on. So there you can produce your first sugar, which is relevant in biosynthesis. Today we talk about catabolism, remember. And you can also use an epimerase where you just move the hydroxyl groups to a different position. So these are of course stereoactive substances, and if you have it at the other position, it's a different substance, and then it's called xylulose. So again, you don't need to remember by heart these sugars. And what you have then is you can have transcatolases and transaldolases. Transcatolases transfer C2 units, transaldolases transfer C3 units. And if you, for example, use here a transcatalase with ribose 5-phosphate and xylulose 5-phosphate as a substrate, which are both C5 units, then you generate a C7 and a C3 unit. Glycerinaldehyde 3-phosphate, this is something you know. It's a sugar, by the way, by definition, because it has a reductive aldehyde group and it has a hydroxyl group. That's the definition. Then you are sugar. And this is called acetoheptulose 7-phosphate. Cool name, but it's of course something you don't need to remember. So this is then, you just can generate with this pathway, with its reversible non-oxidative reaction, you can generate a pool of whatever sugar you want at the end. And this is reversible, so you don't need energy for that. You can just shift. If you say, ooh, I need more whatever, erythrose 4-phosphate, you can use this pathway to produce it. That's the key here. Here are the details how this is happening. This is not important for you. It's important to remember it's a C2 or C3 transfer. So in the pentose phosphate way, in non-oxidative part, we have isomerases and epimerases, and the transcatalases and transaldolases. So with the transcatalases and the transaldolases, we change the number of carbons by transferring C2 or C3 units. That's just the summary what I said. In the oxidative part, I generate 2 molecules of NADPH, and I come from a C6 sugar and generate a C5 sugar. The reversible pentose phosphate pathway, I can generate a pool of C3 and C7 sugars. I can also use that to feed in a sugar not different from glucose, what we discussed before, using the reversible pentose phosphate pathway. Why do you have that? That's what I said. If you have a non-hexose and want to do glycolysis, you can feed it in. You can go then the reverse direction. You can convert sugars, and you can generate NADPH for anabolism. Remember, in anabolism, you also need reductive equivalence. That's what you can do here. And of course, and that was our starting point where we had no aldolase. If I have enzymes missing in my glycolytic pathway, like the aldolase, then I can use this pathway to go around. And you remember I have generated glycerinaldehyde 3-phosphate here. in this pathway. And of course, and this is without an aldolase. So if I have now this glyceronaldehyde 3-phosphate, and this is also here, if I have now my glyceronaldehyde 3-phosphate, I can go the next steps in my glycolysis pathway. Yeah, that's the cool thing here. And welcome to heterofermentative lactic acid fermentation. This is exactly what's happening here. So I have my glucose. The starting point is always the same. My hexokinase is the first step. So I have glucose 6-phosphate. And now I have these two subsequent oxidative steps. And one of them is an oxidative decarboxylation. And I produce ribulose 5-phosphate. I have then an epimerase, and you remember an epimerase was this hydroxyl groups for change, and I produce xylulose 5-phosphate. Do I expect you to learn these structure formulas of ribulose and xylulose by heart? No. This is really just learning by heart. Where is now the hydroxyl group located at ribulose and xylulose? Where is it a keto group? It's an aldehyde group. This is just learning by heart. It's important that you remember that you come from a C6 sugar and you come from two oxidation reactions, and one is an oxidative decarboxylation that you produce here, two NADPHs, and that you produce a C5 unit. And of course, you can convert these C5 units into other sugars using epimerases and isomerases. Now we have our xylulose. If we have a pentose, we can directly, of course, go to this xylulose. So here, if I have a pentose kinase, if I feed a pentose, if I have a pentose kinase, I produce the pentose phosphate. And depending on what pentoses I have, with epimerases and isomerases, I also can end up with xylulose phosphate. So this is something where I don't need an oxidation in between. If I feed in a C6 sugar, I need an oxidation reaction in order to come to this ribulose, because I need to do a decarboxylation to have come from C6 to C5. And first of all, I need to produce a carboxyl group, and this is an oxidation. And then I have a decarboxylation. And if I don't do an additional oxidation, I wouldn't have a sugar, because then I would end up with a hydroxyl instead of a keto or aldehyde group, and this wouldn't be a sugar. So I need to have two oxidations to come from a C6 sugar to a C5 sugar. Yeah, so this means two oxidations. So here I'm at my xylulose, and if I'm here, the next enzyme, it's called phosphoketolase. And you remember in the glycolysis, we have an aldolase. An aldolase splits C3 units. And here we have a ketolase, and the ketolase, remember the transketolase, transaldolases, it splits at C2 unit. So this is what the phosphoketolase does. It splits a C5 sugar, xylulose, into a C3 unit, which is glyceraldehyde, and a C2 unit, which is acetyl phosphate. And here this acetyl phosphate comes from the end where we have already the phosphate. And this here, this glyceraldehyde 3-phosphate is produced in the step where we have the splitting up and a phosphate is being transferred. So the phosphoketolase produces glyceraldehyde 3-phosphate. This is our known glyceraldehyde 3-phosphate and acetyl phosphate. This is acetate. And now glyceraldehyde 3-phosphate, of course, we can go further. We can do oxidation. We can produce pyruvate at the end. And of course, then we could then reduce to lactate. So this is what we have seen already in homofermentative lactic acid fermentation. The steps here are the same as in glycolysis, as in the Embden-Meyerhoff-Parnasse pathway. We produce pyruvate. At the end, we produce also an NADH here, and then we have lactate. Now, what happens with this acetyl phosphate? It depends. So we can, with acetate kinase, we can produce ATP and acetate. You remember, the phosphate here is bound at a carboxyl group. So it's a phosphoanhydride bond. This is energy rich, and this can be used for substrate level phosphorylation. So we can recycle this energy into ATPs. So we have the ATPs which have been brought in, we have taken out. Or what we can do is we can then reduce in two steps the acetate into first step acetaldehyde, second step ethanol, and then put the NADHs on that. And you already feel that this we need if we are here. But in this case, you cannot produce ATP. So this is heterofermentative lactic acid fermentation. Heterofermentative because you don't just produce lactic acid, you also produce other things like acetate and or ethanol. So this is something we do in the lab course. We will analyze, we will use different sugar as sources, we will use different bacteria which do homofermentative and lactic acid fermentation, and then we will analyze the products. What we will do is we will do then an HPLC analytics and looking at what's happening there. So this is just an outlook for the lab course. Now if I start with my C5 sugar as a C source, there is no need for an oxidation step at the beginning. I need an oxidation if I come from a C6 sugar and I want to go to a C5 sugar. And if I come already in with a pentose, I might need to rearrange in order to arrive at cellulose, but this is not something where I need an oxidation because I already start with a sugar. So in the case of pentose, of course, I will have a phosphorylation. That's always the case to keep that pentose in the cell. And then I have depending on the pentose, I might need some epimerases or isomerases to arrive at cellulose phosphate. And then the next step is again my phosphoketolase. I have my lactate production starting from glyceraldehyde 3-phosphate. And now you see the reduction equivalents which are produced here are being recycled. So what I can do is now I can really use this phosphate here and transfer it to ATP via substrate level phosphorylation. So I have recycled the ATP which have been used here and producing acetate. So if I feed in a pentose, I would expect in a heterofermentative lactic acid fermentation pathway production of lactate and acetate. And my net balance here is I have my pentose and I produce lactate, acetate, and 2 ATP. I produce no carbon dioxide. And this is, of course, something we will then have a look on in the lab course. The ATPs we can't measure, but we measure the production of the metabolites and we measure the production of gas. Now, what happens if I start with a C6 sugar? I already said it. If you want to convert a C6 sugar into a C5 sugar, you have to oxidize. There is no other possibility. So we have 2 oxidation steps, and one of them is an oxidative decarboxylation, and we end up then using an epimerase, then add cellulose. We do the same. We are here with my glyceraldehyde 3-phosphate, pyruvate lactate. So this is straightforward. I directly convert it. So this is the part which is the same in homofermentative lactic acid fermentation, and of course also in the heterofermentative lactic acid fermentation. If I start with a pentose, it's the same. Now what happens with my acetyl phosphate? The problem here is that I have produced 2 reduction equivalents. So it's NADPH. This is not, you cannot directly convert NADPH into NADH. However, the let's say the redox potential in your cell, whether you have NADH or NADPH, it's the same. So overall, to get the overall redox potential in your cells with the same level, what you need to do is you need to get rid of these reduction equivalents, depending on how much anabolism you have downstream. But if it's just the catabolic, for the pure catabolic reaction, you need not really to recycle these NADPHs, not these molecules, but you need to take the same amount of NADHs then away in order to have a balanced reaction. So you cannot go to acetate in this case and secrete it and recover the energy from the phosphate. What you have to do is you go to a next reaction in order to be able to produce then to get rid of the electrons. And the intermediate here is acetyl-CoA. So you see that CoA is bound. The phosphate is leaving as an inorganic phosphate. Remember, we had the reverse reaction where we came from acetyl-CoA. We have produced from pyruvate by an oxidative decarboxylation step if we go into the citric acid cycle. Now we go the other way around. The acetyl-CoA we use then with the acetyl-CoA reductase and produce acetaldehyde. So here one molecule of hydrogen is being consumed. And at the end, we reduce acetaldehyde with the alcohol dehydrogenase to produce ethanol. This is the famous alcohol dehydrogenase, ADH, producing ethanol. And of course, here, by the way, acetyl-CoA reductase is called after the forward reaction. The alcohol dehydrogenase ADH is called after the reversible back reaction. And there we consume then 2 NADHs. So the balance, of course, remembering that this is not a real balance because we produce here NADPHs and we consume here NADHs, and that's not the same. But concerning reduction equivalence is if we start with an hexose, we produce also lactate, we produce ethanol and carbon dioxide. In this case, here the carbon dioxide is produced and just 1 ATP. So the energy efficiency for a hexose is lower. So in other words, a hexose, if you have a bacterium which grows in an environment where you have mainly hexoses, using the heterofermentative lactic acid fermentation pathway, it's not so smart. So there, a homofermentative lactic acid fermentation pathway makes more sense. If you have a pentose, it might be wiser to use the heterofermentative lactic acid fermentation pathway because you can produce easily 2 molecules of ATP there. So you see, it depends on the environment, and because you would say, why the hell should an organism lose so much energy with heterofermentative acid fermentation? Yeah, if you sit in milk and if you're used to having hexoses around, it's wise if you go for a homofermentative pathway. But if you are in an environment where you're mainly exposed to C5 sugars, heterofermentative lactic acid fermentation makes sense. And lactic acid bacteria have a vast diversity of capacities, what sugars they can use. So not all pentoses, for example, can use by all lactic acid bacteria because you need, of course, the enzymes to be able to convert it. But this is the cool thing about lactic acid bacteria, that they have this vast metabolic diversity with respect to fermentation of sugars. So that's their hallmark. Now also the heterofermentative lactic acid fermentation pathway is used for food production. So kefir is produced using this pathway. And therefore this is the taste is different, of course, richer using heterofermentative lactic acid fermentation. Okay. Just summarizing the key points of lactic acid fermentation, we have the homofermentative pathway. So we need the aldolase of the Meyerhoff-Parnasse pathway. Usually use a C6 sugar as a C source. And by the way, lactose in milk is just beta 1,4 linked galactose, which is a C6 sugar, and glucose. And the major part of food products are exploiting this pathway. Most lactic acid bacteria indeed have this pathway, like Lactococcus, Streptococcus, and some Lactobacillus species. But the heterofermentative pathway, it's cool if you grow on C5 sugars. In this case, you use the so-called phosphoketolase pathway. We have this phosphoketolase in between. And here our products are lactate, acetate, and ethanol. Of course, the composition depends both on the strain and the substrate. We will do that in our Applied Microbiology lab course. Only some food products are produced using this pathway, for example, kefir. In general, these bacteria don't have an aldolase, and so you could, if you have a lactic acid bacterium, by using sequencing, looking whether they have a functional aldolase, you could already predict whether they are homofermentative or heterofermentative. Examples which are heterofermentative are Leuconostics and some Lactobacilli species. Okay, it's a bit more than just glycolysis and adding another enzyme, which is homofermentative lactic acid fermentation. But at the end, it's not so complicated. You just oxidize at the beginning instead of the end and do a bit of a rearrangement, and more or less that's it. So therefore, also heterofermentative lactic acid fermentation and the pentose phosphate pathway is not a complete secret anymore to you, hopefully. And I want to thank you for your attention and have a nice day.